Thanks. Hi, I'm Sarah Heilbronner, and I've spent my career focused mainly on evaluating anatomical connectivity using track tracing in animal brains. So I want to talk about relevant topics in this domain, and sort of, I won't bring up MRI, right? I'm going to focus on connectivity mostly. And so first of all, one underappreciated feature of cortical-cortical connectivity that we've actually known for a long time but not studied in detail is how patchy it is. That is, you put an injection into a particular region and look at the downstream or upstream labeling, and you don't see uniform label across the entire region. The resulting pattern occupies just a portion of the cytoarchitectonic area or even sub-area, and sometimes a patch actually appears to cross the areas. We don't understand how these patches work. It was clear, it's clear from all of the previous presentations, this principle came up. But in sensory motor cortices, we might be comfortable calling these columns. In other parts of cortex, maybe not, depending on your view. But for example, we don't know whether adjacent patches in one region project to adjacent patches in the downstream region, or whether some other pattern, which I've sort of outlined here, is obeyed. And the reason for this gap in the literature, I'd love to talk to anybody, literally anyone in the room about after this, but I'll assure you we're making headway on solving it now. But to me, I think this is probably a very important foundational organizing feature that explains a great deal of function. And indeed, we, in my lab, noticed patches of label after injections in the posterior medial cortex, around the medial orbital sulcus and the orbitofrontal cortex, and then simultaneously recorded in both regions while monkeys performed a reward-based decision-making task under risk. And then we segregated neurons according to whether they were inside the part of the orbitofrontal cortex that connected with the posterior medial cortex or not. And you can read this paper from Maya Wang. She found fairly dramatic functional distinctions such that the connected part of the orbitofrontal cortex seemed to transfer value information to the posterior medial cortex, which then itself added spatial information, but this was not true of the unconnected part of orbitofrontal cortex. Now, you might say, you know, this is just one circuit, and you might have selectively recorded from inside one cytoarchitectonic area and another, it might be biased, and so really this is actually about cytoarchitecture. And so I want to highlight these studies, which haven't come up yet. These are all from the mice. The techniques have existed in mice for a little bit longer to let us evaluate this in a much less biased way. These are results from 3 different papers, not mine, all in mice, all involving clustering from sampling methods that can be performed at high spatial resolution. And this one at the top is top right is connectivity based. Bottom left is connectivity based. Top right is transcriptomic based, and bottom right is actually functionally based on electrophysiological signals. And you can see that sometimes the clusters correspond to cytoarchitectonic areas, but often they don't. And not just because they make smaller units than areas, which you could imagine, especially layers, might differ, but sometimes they make larger ones. Sometimes they even make larger ones that are not spatially contiguous with one another. So I think, like many of my colleagues, I envision many levels of useful organization that provide utility in explaining brain function and dysfunction, which is the ultimate goal, right? Some smaller than the area, some larger, and some that may be spatially non-contiguous. And so for me, this is territory that I'm familiar with from my studies not of the cerebral cortex, but of the striatum. Of course, the striatum is a subcortical structure, and you can kind of parcellate it into the core of the nucleus accumbens, the shell of the nucleus accumbens, and the dorsal striatum. But the truth is that the core of the nucleus accumbens does not have a clear boundary, not a sharp boundary with the dorsal striatum, although it does with the shell. And then within the dorsal striatum are islands of striosomes inside the matrix that, although spatially non-contiguous, have things in common. On top of that, connectivity obeys gradients, but also has patchy features, meaning a given cortical region projects to, can have patches of areas that have projection and areas that don't. Anyway, we are very comfortable with these levels of organization within the striatum, right? And we're able to seek to understand how they interact functionally in health and disease. And I would hope to make that analogy and do the same for the cerebral cortex.